This session is concerned with molecular assemblies and molecular architectures, which, as you will see, relies heavily on the use of non-covalent interactions. Chemistry, of course, as we recognize, is primarily concerned with the organization of atoms in space, and they use two kinds of interactions to do so. One is the covalent interactions, the other you might call non-covalent interactions. Now, in the introductory portion of this session, I would like to just highlight the differences and the manners in which these two interactions interplay to create complexities and functions. Of course, a lot of our inspiration for this is derived from the way biology or life forms itself work. So as we know, as I was mentioning, chemical structures are made up of a variety of bonds, and covalent bonds are those that are place atoms spatially in a manner which rigidifies the relative position of atoms. For instance, if you take a small molecule like benzene or cholesterol, the relative positions of these atoms are fixed and allows for little variability. The variability comes from the ability of these atoms to move or vibrate. Now, there are other kinds of molecular systems, such as, for instance, polyethylene, which is another molecular system, which is a string of carbon atoms, but despite the presence of covalent bonds that link these carbon atoms together, they have flexibility around certain bonds to adopt varying shapes and sizes. So polyethylene, for instance, can adopt a variety of shapes and sizes. And a lot of the function utilizes the ability of these atoms to move around more than the relative stiffness of these atoms with respect to their positions. Now, in contrast to covalent interactions, non-covalent interactions are relatively weak, and importantly, they are also reversible. Now, among the whole range of non-covalent interactions that chemistry utilizes, hydrogen bond is one of them, which of course most of you recognize. Similarly, electrostatic interactions between oppositely charged ions. And then there are pi stacking interactions, which occurs when you have extended p orbitals which have lone electrons in them, such as this. We have extended pi conjugation in this, an aromatic ring system, which has interactions with other aromatic ring systems in this fashion. And in case the aromatic systems are of opposite electron demand, as you will see in some of the cases we have done, you get charge transfer interaction, which is a variation of this kind of interaction. In addition, you also have solvophobic exclusions. Among these non-covalent interactions, there are some of them which are highly directional, and some of them that are not so highly directional, and the functions and structures that control chemistry often rely on directional interactions much more so than on non-directional interactions, such as dispersion interactions, which are what is operational in solvophobic or hydrophobic interactions. Now, in contrast, so in comparison to covalent bonds, which are about 80 to 120 kilocalories per mole, non-covalent interactions are weak, as I mentioned, 1 to 7 kilocalories per mole, and often these are made, formed, and broken during the time scale of the experiment itself. Very importantly, these interactions can also be destabilized by external stimuli in the system, such as pH, temperature, and so on. And the reversibility of these interactions is often crucial to the kinds of functions that we observe in biology. Now, we are all familiar, of course, with the importance of such weak interactions. In this case, these are directional hydrogen bonding interactions that hold the two strands of DNA together. And you will notice that in each case there are multiple interactions, multiple hydrogen bonding interactions between substances, which I'll come to later. And so the beautiful structure that we are familiar with has its origins in the existence of these weak reversible interactions. And this, as I mentioned, the reversibility, the ability of these strands to actually open up is key to the replication process in DNA itself. Now, similarly, weak interactions are also particularly important in catalysis and transport and so on, which are the primary functions of a whole lot of polypeptides, proteins in our living system. And here, of course, the structure, that is, here the interactions, as opposed to being from two independent molecular entities, the interactions stabilize the conformation of a long macromolecule, such as proteins. The interactions in these cases, in order to stabilize these folded forms, are often between segments that are non-adjacent. And as Professor Moore points out in one of his reviews a few years ago, that's critical to the cooperative nature of the folding process itself. And what you mean by that is the interaction between these two subunits here actually plays the next unit that is going to interact in the right proximity for this interaction to happen. Now, once the secondary structure of the protein is formed, it again folds to form this sort of tertiary structure, which is often driven by the non-directional interaction, which tends to expose the more hydrophilic regions of the protein to the outside surface, which lowers the energy of the system. So a lot of the function of these proteins directly depend, catalysis for instance, directly depend on the ability of such complex folded structures to hold the constituent entities that are going to react in right disposition so that the reaction can happen. So these examples very clearly demonstrate that the interplay between covalency and non-covalency is very critical to the development of structure and function in biological systems. The recognition of this has stimulated extensive research, primarily by a refocusing of these objectives by the works of Jean-Marie Lehn many years ago, which helped people to examine these issues more in the context of developing synthetic systems which will exhibit similar structures and functions. There are lots of examples, and I've just chosen to highlight a couple of them. In this session, of course, there will be two talks, one on functional organics by Ajay Ghosh, which will primarily deal with how to use these weak interactions to create manifolds of structures which exhibit specific function or material properties. The other talk is by James Novick, who will examine the issue of foldamers, primarily utilizing hydrogen bonding interactions to stabilize a certain conformation of a synthetic macromolecule. Now, I'll just examine two systems where, the presence of multiple interactions, weak multiple interactions, can enable dramatic increase in the formation constants of complexes. For instance, pioneered by Meyer and co-workers, the presence of this particular unit, as you will notice, has ability of forming 4 hydrogen bonds, as you've shown here. And the relative disposition, the relative positions of these atoms are fixed by covalency. And the ability to form 4 hydrogen bonds increase the formation constants enormously compared to a single hydrogen bonding. And of course, it doesn't scale as 4 times the strength of a single hydrogen bond. I was thinking of a parallel. When we were kids, we play with other kids. When you hold somebody's hand and the other person is trying to release it, he opens one finger at a time and will have an incredible amount of difficulty to get out of it. It's because the timing of opening of all the 4 bonds are not the same. So in a sense, when you have four bonds that are holding things together, the simultaneous breakage of all these 4 bonds is essential to actually decouple this interaction. And that becomes harder. So an extension of this, of course, is that if you take a system that is capable of forming 4 hydrogen bonds and link them up with a covalent linker, then immediately you come to the possibility of generating what would be called now supramolecular polymers, where the connectivity between the individual units are no longer covalent linkages, but they are non-covalent reversible linkages. And I noticed that Craig will be talking about this, and I also noticed his fine examination of the importance of thermodynamics and kinetics and the interplay of these factors in governing the final properties. Now, what is interesting about these class of systems is that unlike covalent polymers, where each repeat unit, for instance, is linked by a series of covalent bonds, in non-covalent interactions, these are hydrogen bonds or multiple hydrogen bonds that hold them. What is unique about this kind of structures is that the properties of the system is strongly correlated with several of the properties of the solution itself. For instance, as you increase the concentration of the monomer units in the solution, the length of the chain actually continuously increases. Okay, that's a consequence of the implication of the concentration on the association constant itself. Now, more importantly, one can also vary temperature to cause these macromolecular organizates to break up into smaller and smaller pieces. So you can see that there's a dramatic difference, for instance, in a property such as viscosity as a function of temperature in these systems, because disconnections cause the population of smaller and smaller oligomers to increase as you go on, as you increase temperature. Another interesting hypothesis of the importance of non-covalent interaction is in the formation of these so-called molecular footprints or molecular fossils. Pauling many years ago wrongly postulated, but it triggered a lot of work in this area, and that is, he postulated that the interaction or the ability for an antibody to recognize an antigen happens primarily by the conformational adaptation of this macromolecule around an antigen or a template. And once the conformation is fixed, its ability to recognize this antigen is dramatically improved. So it's, in a way, it's reorganization of the macromolecule so as to be able to recognize the antigen. Now, what it triggered, of course, is the imagination of chemists to utilize this idea of organizing non-covalently molecular entities that can be fixed in space by polymerization around a template, such as so, and then polymerize. These double bonds can be polymerized to be completely fixed, and once you polymerize, you pluck out this non-covalently bound system, and you create a topological cavity which has elements that will primarily recognize the template for which it was developed. Now, this process is called molecular imprinting, and it's more like a footprint, a molecular footprint, in this nice cartoon depiction in the article by Zimmerman. What I would like to sort of draw your attention to is a much more interesting examination of this idea, which is much closer to the Pauling hypotheses. And here what is done is you take a large macromolecule which has several points which can interact with a template, and you lock in the conformation of this macromolecule around the template, and then you fix the conformation of this locked-in macromolecular conformation by pinning it down to a substrate by covalent linkages. Now, what this does is it fixes the conformation of this polymeric chain in such a fashion that it provides limited ability for these recognition units to move around. So once you pluck out the template, these units that can recognize this template are in the right disposition so as to pick up these kinds of molecules much favorably over the ones that it's not imprinted for. In this example, it was glucose versus fructose. I would just tell you, just in the last 2 minutes, I mean, by way of introduction, I just wanted to examine the differences between these. But in the last 2 minutes or so, I'll just tell you our own efforts in this area of trying to control conformation of polymers by using weak non-covalent interaction. And we have excluded hydrogen bonding on purpose because we felt that's the most examined system. So we used a variety of other forces to look at two kinds of systems. One where we look at charge transfer interactions and metal ion chelation of a relatively flexible loop so as to bring the aromatic rings in right registry to form this complex. Another example is a system where we take a large macromolecule and then use a small molecule which interacts with two separate segments, adjacent segments on the macromolecule, so that it brings these two units in right registry to form the complex. we examined these like most chemists do. In fact, this donor-acceptor interaction was something that was examined by Iverson and others. The difference we have is that we use oligoethylene glycol units, which are much more flexible, but can be made constrained by interacting with the metal ion. We examine these things by a variety of techniques. By way of introduction, we use nuclear magnetic resonance spectroscopy and absorption spectroscopy. And of course, absorption spectroscopy simply tells you that the intensity of this charge transfer band, which happens as a result of this electron-rich and electron-deficient aromatic ring coming in close proximity with each other, developing a band that has an absorption visible. The intensity of this band is a reflection, is of course obvious by looking at it, is a reflection of the extent of this interaction that is present in the system. Similarly, in NMR spectroscopy, we simply examine the relative position of the peak that belongs to one of these units, and this relative position, in a sense, gives us an idea of the extent of this interaction that has happened in the system. The position turns out to be a mole fraction weighted average of the forms that are free and complex. So we can modulate the extent of interaction in these cases by the presence of metal ions, as very clearly evident in these spectral signatures. So in the presence of an appropriate alkali metal ion, it constrains the conformation of the loop, thereby bringing these two in right registry so as to be able to form these interactions that give us signals spectroscopically, as well as lead to folded conformations of the polymer. We go on to ask questions such as what is the length scale over which such organization can happen, and we do it by primarily comparing spectral variations in the polymer with that of the small molecular entities, specifically designed model compounds that allow us to examine the variations. What we effectively see is that the variations of the small molecule components parallel that of the model compounds in a manner so as to indicate that there's a very significant fraction of the acceptor units that are sandwiched between donors, and similarly a very significant fraction of the donor units are actually sandwiched between acceptors. This is something that tells us that the stack length might be very long, but unfortunately the technique is not sensitive enough for us to actually calculate the stack length, because the variations happen to flatten off as the length scale actually increases. In the external added folding, I'll just close with this. The interesting feature here is the presence of this ammonium ion, which is very similar to a metal ion. The ammonium ion now complexes with this loop, brings this donor atom in right registry so as to clasp around these two acceptor units. And this is particularly interesting because what it allows us to do, I'll skip this, what it allows us to do is to do this in a completely reversible fashion. So which means I can take a macromolecule that has a random conformation, for instance, and come in with a unit that simply plugs into it in such a way that it folds the macromolecule in a particular fashion as shown here. The interesting feature about this is our ability to pluck out this at will by utilizing an appropriate agent. For instance, in this crown ether, which complexes with this agent much more strongly than the other one. So all the spectroscopic signals that suggest formation of a folded conformation is completely reversed when you actually take this out. This is an interesting feature, which means if you are able to translate this idea into the solid state, you would be in principle capable of organizing it in solution, translating it to the solid state, plucking out the folding agent, and retain the folded conformation potentially in the solid state. Now, my conclusions, of course, is going to be sort of more philosophical in nature. It is evident from the comparison that we've made with synthetic chemistry and macromolecules and polymers that weak non-covalent interaction, the balance between covalent and non-covalent interaction is critical to a lot of functions in biology. Now, in my own perception, polymers actually form a wonderful interface between the covalent and non-covalent chemistries. And the reason I say that is, of course, if a particular function, I mean, when you think about it, biology has selected long macromolecules for a specific reason. The entropy costs associated with developing a function, if you think about it, macromolecules have a very large number, let's say 10,000 plus atoms in them. The ability to bring, or the entropy cost to bring such large number of atoms together in a particular spatial disposition is very high. The way nature works up that, or the way it compensates for that, is to tie down these atoms along a polymer chain, to form a long polymer chain, giving it adequate mobility to reorganize, at the same time reducing the entropy costs in bringing such a large number of atoms to perform a stable function. So it's an interesting way. So as we learn more about this atom jugglery, if you will, the way in which spatial and temporal locations of atoms is of course the key to being able to understand and mimic a lot of the structural and functional uniqueness of life forms. One of the hardest problems, yesterday's talk by Professor Arkin, reminded me when I was looking at the manner or the cooperativity with which these organisms, these unicellular, unthinking organisms, organized to sort of perform a particular function, makes you wonder that the distinction between